All right, now let's talk about solution concentrations. You're probably not very familiar with the words that you see in front of you. Hypotonic, isotonic, and hypertonic are typically new to students in biology. These are comparative terms. When you see the word hypotonic, it is telling you that whatever we're talking about has a lower concentration than something else. So if you're saying your solution is hypotonic, so the solution is this area outside of the cell. So we'll say this is the cell, and here's the outside of the cell, it's the solution. The solution in this scenario is hypotonic because it has a lower concentration of substances than inside the cell. And you can plainly see that inside the cell there's more of this green dot material. Okay. Going to the other side of the spectrum is hypertonic. Hyper means higher. Think of if you're very hyper, you've got a very high level of energy. So hypertonic solution has a higher concentration of substances dissolved in the solution than inside the cell. And again, you can look inside the cell and it's like, okay, yeah, I can see that it has some, but there's a lot more outside. That makes the solution, this area outside of the cell, hypertonic. And the one in the middle, obviously, if you've got a situation where the solution concentration is lower than the cell and a situation where it's higher than in the cell, there's got to be one that's in the middle. So iso means same, meaning that the concentration of the solution is same or very similar to that of the concentration, or to that of the cell. The concentration in the cell is similar to the concentration in the solution. Now, there's a reason that this matters. When you have differences in concentration, this is a concentration gradient. So we've already been talking about that with cell transport. Here you have a concentration gradient where the concentration of particle is higher inside the cell than outside the cell. And here you've got the concentration is higher outside the cell than inside the cell. But again, that's a gradient. So we want to talk about how are these particles are going to move. Well, what if I told you that these particles cannot go into or out of the cell? They are stuck where they are. Or at least they can't move very rapidly. And in nature, we want to find some kind of balance. We want to be able to find a way to have relatively equal concentrations both inside and outside the cell. So if these particles cannot move into or out of the cell, then something else is going to have to move, and that something else is the water. So let's look at this picture where this is essentially showing the same thing, but I'm going to show you the isotonic scenario first. So in an isotonic solution, this is showing a red blood cell immersed in water. So it's showing you the water, but then also notice there's all these other molecules in here. We don't know what they are. It doesn't even matter at this point. But those molecules are dissolved in the water, and there's also some inside the cell. Since we have pretty equal concentrations of those molecules inside and outside the cell, water is going to move into and out of the cell at very equal rates. So it's not like water is going to rush into the cell to equalize the concentrations, and it's not going to rush out of the cell either. It's going to move into and out of the cell at about the same rate. And we'll look at that some more in a minute because there's a word for that. Here you've got a scenario in which here's this red blood cell that is submerged in water, and within that water you have some dissolved substance, but you have even more dissolved substance inside of the cell. And remember, I told you that that molecule, that substance, cannot leave the cell. So somehow we want the concentrations inside the cell and outside the cell to be more similar. And the only way to do that is for water to move into the cell. So water will actually rush into the cell. And as you can see, this cell is very plump. It has swollen up a bit because water has rushed in. Notice water also comes out, but the idea is that more water has gone in than come out. That's why you see net water gain. So this situation where you have a hypotonic solution, remember the hypo means low. So this solution that the red blood cell is in has a very low concentration of these molecules. The opposite is true here. In the solution in this beaker, the concentration of these molecules is higher. Therefore, this cell is in a hypertonic solution. And if that happens, remember, those molecules aren't going to move into the cell, so water will have to move out of the cell. And in a hypertonic environment, a cell will shrink. So these are conditions that you do need to be very familiar with and be able to describe. Hypo, it has a very low concentration outside the cell. Therefore, water will rush in order to try to equalize those concentrations. Hyper is higher concentration outside the cell. Remember, when we're talking about these higher concentrations, we're talking about higher concentrations of these molecules, not higher concentration of the water. Okay, so we're going to talk a little bit more about that in class, so try to follow along for now. But remember, we talked about isotonic. So again, here's our isotonic solution where the concentration of these molecules in the solution is very similar to the concentration in the red blood cell. And again, notice water will move into the cell, but it will also move out of the cell. So in this case, in an isotonic environment, you're going to have pretty much no loss or gain of water or size or anything like that because the same amount of water is leaving the cell as is coming in. This is called dynamic equilibrium. Okay, so I want you to really print out or look at the skeleton notes and go through all of the vocab words that I have talked about in the last two videos. Make sure you understand all of those vocabulary words and are able to use them, articulate them, and compare all of them. All right, we're going to talk more about this, and we might be able to get our hands wet with some examples as well.